Escherichia coli are added. Here also Escherichia coli were added or added. Okay, E. coli allowed them to grow for several generations. Here the E. coli are also allowed to grow for several generations. After these several generations of growth of E. coli, the DNA of the E. coli isolated from this medium, subjected into centrifugation. What is the name of the centrifugation? Cesium chloride density gradient centrifugation. It was found by Stock and Michelson for this experiment. So in which they have found a band which is formed here itself. This DNA band is known to contain heavy nitrogen, that is called N15. DNA from this E. coli of this culture medium isolated and subjected into centrifugation and got the band. Such a band is known to contain N14 nitrogen. So by which they won't come, they didn't explain anything else, they didn't find anything else, which is the right method of DNA replication, either semi-conservative or conservative or dispersive. Nothing can be explained up to this experiment one. Then what they have done? They have done the second experiment. You see here, it's a second. Here from the heavy culture medium, only one E. coli was taken and subjected into a medium which is known to contain only N14, that is ammonium chloride. Ammonium chloride is there as a nutritive substances in which the nitrogen is N14 nitrogen is only there. There is no any N15 in this medium. Is it clear? So before introducing the E. coli into this medium, what medium? NH14 medium. E. coli taken from NH15. In NH15 in the E. coli is known to contains nitrogen, that is heavy isotope. Heavy isotope nitrogen is known to found the DNA which is isolated from the culture medium A. So before introducing into it, the DNA is taken and centrifuged and observe the DNA strand formation. 100% heavy nitrogen found in this. So after they have added into this E. coli first generation, that is first replication. Okay, first replication. You see, this is the original N15 DNA strand isolated from the E. coli. When it is allowed into N14 medium and grown for first generation, from the first generation of E. coli, DNA was isolated and centrifuged and observed the band formation. One it is heavy, another one is light. Here one is heavy, another one is light. The heavy light I am mentioning now, the heavy refers to nitrogen 15. Nitrogen 15 isotope is present. Here N14 isotope is present in the first generation. Then they allow the first generation to produce the second generation within 20 minutes. The duration of the replication of E. coli is only 20 minutes. So within 20 minutes, they produce the next generation. The DNA of the second generation isolated and centrifuge, and they observe the strand formation like this. So here one strand as such present. Here also one strand, that is N15 is present. All the remaining strands are N14 because the medium is N14 medium. E. coli taking the nitrogen from NH4Cl medium. So DNA, newly synthesized DNA is N14 normal isotope. Nitrogen normal isotopes are there. And then the second generation allowed to produce the third generation. Whereas DNA taken from the third generation of E. coli and subjected into centrifugation, one heavy strand is maintained. Here also one heavy strand is maintained. All the remaining are light strand, that is N14 strand. Then fourth generation, one strand is maintained. Here also one strand is maintained. All the remaining strands are the N14 strand. So after the fourth generation, these two scientists have come to a conclusion that semi-conservative method of DNA replication is only right because one strand is continuously maintaining in the forthcoming generation. So there is no chance for dispersive, there is no chance for conservative. One strand as such is maintained, by which these two scientists have proved that semi-conservative replication of DNA is right. Any doubt? Okay. So I did well. Move to the 0 hour. This is a time for you. You take only 2 minutes. Semi-conservative replication was proposed by anybody?
Watson and Crick, sir.
Good. In the year
1953, sir.
Okay. Excuse me. Any doubt in the experiment? Any doubt, please? If you have any doubt in this experiment, please bring to my notice. Is it taught to you in the NEET Pinnacle team? Any one of you? Hello?
Yes, sir.
Yes. இப்போ clear-ஆ இருக்காடா இல்ல இன்னொரு டைம் ஏதாவது எக்ஸ்பிளைன் பண்ணணுமா?
Short recap, sir.
Recap. Okay. ஒண்ணும் இல்ல. ரெண்டு என்ன எடுத்துட்டாங்க? There are two medium. Okay. This is the medium, another medium. Here N15 is the nitrogen source. Heavy nitrogen is added in NH4. Here only 14. Then added the E. coli in this both culture medium. Okay, allow them to grow for several generations. After the several generation of growth of E. coli, the DNA of the E. coli isolated from this medium and centrifuge and observe the band formation here. The band is known to contain N15. N15 is the heavy nitrogen. N14 is the normal nitrogen. As such, in this side, the DNA of the E. coli isolated from this culture medium and subjected into centrifugation, cesium chloride density gradient centrifugation. The band is formed like this. Observe the studied the band. It is known to contain N14. ஏன் நைட்ரஜன் எடுத்துக்கிட்டாங்க யாரா சொல்ல முடியுமா? Ammonium chloride is used as a source of nutrients. Why? Anybody can you? ஏன் நைட்ரஜனை யூஸ் பண்ணாங்க? அம்மோனியம் குளோரைடை யூஸ் பண்ணாங்க ஏன்னா நைட்ரஜன் வெரி எசென்ஷியல் ஃபார் நியூக்ளியோடைட்ஸ் ஃபார்மேஷன். Is it clear? In a nitrogenous base, nitrogen is a major component, no? So nitrogen is very essential for replication. So that they have chosen ammonium chloride as a nutritive substances. This is a first experiment. Second, என்ன பண்ணாங்கன்னா இந்த N15 கல்ச்சர் மீடியத்தில் இருக்கக்கூடிய டிஎன்ஏ இருக்குல்ல, ஐ மீன் E. coli, அந்த டிஎன்ஏ எடுக்கிறாங்க. அந்த டிஎன்ஏவில் என்ன இருக்கும்? N15 is more. It is taken, the E. coli is taken and poured into the N14 culture medium. N14 culture medium introduce பண்ணாங்க. Allow them to grow for the first generation. After the first generation of the E. coli, the DNA taken from and subjected into centrifugation, என்ன வந்துச்சு? Hybrid DNA வந்துச்சு. The first இருக்குல்ல, இது before introducing into the NH14, the DNA of the NH15 medium இருக்குல்ல, அதை centrifuge பண்ணதுதான் இந்த 0. ரெண்டுமே ஹெவி ஸ்டாண்ட் தான் இருக்கு. ஸோ ஹெவி ஸ்டாண்ட் உள்ள E. coli கொண்டு வந்து லைட் மீடியத்தில் யூஸ் பண்ணும்போது ஃபர்ஸ்ட் ஜெனரேஷன்ல ஒரு ஹெவி ஒரு லைட் வந்திருக்கு. தென் சேம் E. coli allowed to produce the second generation. After the second generation is formed within 20 minutes. So after the 20 minutes we have to take the E. coli and isolated the DNA subjected to the centrifuge. பண்ணோம்னா இது ரெண்டும் டிவைட் ஆகும், இப்படி டிவைட் ஆகும், இது இப்படி டிவைட் ஆகும். டிஎன்ஏ ரெப்ளிகேஷன் ஆகும்போது இது நியூ ஸ்டாண்ட், இது அனதர் நியூ ஸ்டாண்ட். இந்த ரெண்டு நியூ ஸ்டாண்டுக்கும் சோர்ஸ் ஆஃப் நைட்ரஜன் எது? N14 தானே. ஸோ இதுவும் N14 ஸ்டாண்டு, இதுவும் N14, இது ஆல்ரெடி N14. ஸோ N14 இருக்குது. ஒரு ஸ்டாண்ட் இஸ் மெயின்டைன், த ஹெவி ஸ்டாண்ட் இஸ் மெயின்டைன். ஹியர் ஆல்சோ one heavy strand maintained. Then second generation E. coli allowed to replicate for the third generation. Then DNA of the third generation is subjected into centrifuge. ஒரு ஸ்டாண்ட் ஹெவி ஸ்டாண்ட், இங்கே ஒரு ஹெவி ஸ்டாண்ட், ரிமைனிங் எல்லாம் என்ன ஸ்டாண்டு? லைட் ஸ்டாண்ட். எஸ். Did you call me? Anybody did call me? Okay. Fourth generation பார்க்கறாங்க. So by this we come to know, in this side one stand is maintained, here one stand is maintained. So semi-conservative is the right approach in the DNA writing. That's all. Okay. Good.